Hi, I hope you all are fine, and yet I'm back with another topic from you, that is vitamins. So vitamins, as you all have heard, that they are very essential for our body, and sometimes it happens that there is a in your mouth there are ulcers. So small sores they are there, and because of that you go to doctor, then he suggests that you eat vitamin tablets. So vitamins are essential for our body. From that point of view, you can understand that they are very important for our body. And vitamins can be found in a variety of foods, from animal sources like egg, from plant sources, pulses, green vegetables. They are a good source of vitamins. So in this lecture, I'm not gonna talk about all the sources and what diseases are there, though I have attached the diseases because sometimes they're asked, but I'm not gonna talk about the sources and why they are important, not all that thing. But we'll see the biochemical viewpoint of vitamins, that how they catalyze, they help in catalyzing the reactions, how they help enzyme in catalysis of the reactions. Okay, so from that point of view, we're gonna study vitamins in this lecture. So okay, so we'll see just chemical point of view of vitamins, structural point of view, and in what all reactions vitamins are involved. So let's start today's lecture. First, let's start with a discussion of what is cofactor. Now, if there is an enzyme and it is catalyzing any reaction to give an enzyme substrate complex and then enzyme plus products. Now the thing is that this enzyme is already performing a single function, that is, it is increasing the rate of the reaction. Okay, now suppose if there is another component that is required by enzyme. So this is a substrate, and suppose in catalyzing a reaction, enzyme requires X component also. So enzyme cannot perform two functions at a time. It cannot go and bring out that X component also so that it can catalyze a reaction. So there comes the role of cofactors. So cofactors are those which help enzymes in catalyzing reactions. So if there is an apoenzyme, that is a protein portion that is inactive. So this is known as apoenzyme. The enzyme that is inactive or cannot function properly, it is known as apoenzyme. And when you add a coenzyme, that is a part of cofactor. We'll see that cofactors has different parts like coenzyme, prosthetic groups. And cofactor is something, if I only talk about cofactor, it is something that helps the enzyme. That doesn't mean that the enzyme cannot complete the reaction by itself, but a cofactor can help it. But a coenzyme and a prosthetic group are needed for enzyme functions. So cofactors, there are different metallic parts also that can help in increasing the rate of the reaction with the enzyme. But enzyme can also function alone without a cofactor. But if there is a coenzyme or if there is a coenzyme, then in that case the enzyme needs a coenzyme to do that reaction, without which it cannot complete the reaction. So there's an apoenzyme which is also known as inactive enzyme, and if there is a cofactor that is attached to that enzyme in the form of coenzyme, right now we are talking, so then there is a complete holoenzyme, and this enzyme can catalyze a reaction. That's why in metabolism, in biocatalytic reactions, vitamins play a major role, and we'll see that how they play this role. Okay, so let's proceed further and talk about the classification of cofactor. As I was telling you, that cofactors can be classified into metal ions. Now these metal ions, sometimes they are, like for example, if I talk about DNA polymerase active site. So while polymerizing nucleotides, you know that nucleotide consists of PO4 negative charges that comes with nucleotide incoming, and if the incoming nucleotide is triphosphate, then it consists of PO4 4 negative charges overall. Okay, now to this negative charges creates a lot of repulsion, and so the polymerization cannot happen effectively. In that case, what happens is that there is an Mg2 positive that is present in the active site of this DNA polymerase, and this stabilizes the negative charges on the phosphate groups so that the reaction can happen fast, and so that there is less repulsion between negative charges. So this is how metal ions can help enzymes in doing this reaction. Though if you remove magnesium 2 positive, then also the reaction can happen, but that point of time there will be so much repulsion that 1 in 1000 times that reaction will be successful, or I can say 1 in 10,000 or 1 in 1 lakh times that reaction will be successful. Okay, next is coenzyme. As I was telling you, that they are needed by the enzyme to complete their reaction. Now they can be co-substrates like NADP positive, NAD positive or NADP positive, or they can be prosthetic groups. Prosthetic groups are those which are actually a part of the enzyme and they are very tightly bound to the enzyme. And co-substrate or coenzymes are those that are loosely attached, like NAD, FAD. Will see that they are loosely attached. They can go their way also. Okay, so prostatic groups usually consist of heme group, like we'll see in RBCs. Hemoglobin also acts as an enzyme that accepts oxygen and also donates oxygen. So in that case, it acts as an enzyme. Hemoglobin itself acts as an enzyme, and the heme group that is present in hemoglobin acts as a prostatic group. So let's proceed forward and let's come back to vitamins. First, let's study about classification of vitamins. Now, vitamins can be classified into either water soluble substances, or they can be classified into your fat soluble substances. So two types of vitamins are there. One is water soluble and another is fat soluble. In this lecture we are gonna talk exclusively about the water soluble vitamins. And water soluble vitamins are known as vitamin B complex. You might have seen this on tablets that you take